Okay, thank you for the introduction and for this opportunity to talk about my PhD project, which is about the acetyl-CoA metabolism and human trophoblast differentiation. So to start with, I will introduce, for those who are not familiar with it, a little bit of background knowledge about the placenta. So the placenta is a unique organ which connects the fetus to the mother, and it's entirely fetally derived, and it is composed of a villus-like tree structure with fingers-like projection, which embeds into the lining of the uterus during pregnancy. And is a unique organ because it has multiple functions which are normally carried out in an adult in different organs, such as gas exchange, metabolic transfer, hormone secretion, and fetal protection. In our lab, we are particularly interested in studying the early stages of placentation. So we know that in humans, implantation occurs at day 7, and we have the blastocyst that hatches, and the outer layer of the blastocyst, called trophectoderm, will start to proliferate and give rise to the cytotrophoblast cells, which are mononucleate and proliferative cells. And these cells will then again proliferate and start to invade the endometrium by fusion. These cells will also form another type of cells called syncytial trophoblasts, which are multinucleated and terminally differentiate. Then the trophoblast cells can also form the extravillus trophoblasts, which are fundamental for the spiral artery remodeling. So understanding the differentiation process is fundamental for us. But why do we focus on trophoblast differentiation? Well, to the placenta to develop and function in a proper way, the trophoblast differentiation is essential in order to provide nutrients and to provide like an organized structure which will connect the mother to the fetus. And when we have an impaired trophoblast differentiation and function of the placenta, this is usually associated with those that we call greater obstetrical syndromes, such as fetal growth restriction and preeclampsia. So on one hand, we want to focus on early stages of placentation, and on the other hand, the other concept that I want to introduce is histone acetylation. So for histone acetylation, we mean all those covalent modification of the histones. So histones are a core protein around which the DNA wraps up. And so when we have this modification, the DNA kind of loosen up a little bit around these histones. And so these genetic regions will be more available for transcription. And so the chromatin will be in a kind of relaxed state. While when these modifications get removed from the histone tails, then the chromatin will be more dense. And so it won't be accessible anymore for the transcriptionary machine. This acetyl-CoA group derives from the acetyl group derives from the acetyl-CoA and is produced in cell metabolism. So do acetyl-CoA levels control trophoblast differentiation? Well, we know that in order to maintain a cell in its stem state, it needs higher histone acetylation levels. And when this histone acetylation drops, the cell will start to differentiate. And this histone acetylation, in order to be maintained, it requires the acetyl-CoA as a substrate. But what regulates the acetyl-CoA is actually the central carbon metabolism. So it's the metabolism that controls cell differentiation via epigenetic processes. So here come our hypotheses. So if we look this pathway that I'm focusing on, glycolysis and all the pathway are mainly involved in the production of acetyl-CoA, which is then fundamental for histone acetylation when it gets in the nuclei and is being used as a substrate, and then this will sustain cytotrophoblast proliferation stemness. And so reduction in this acetyl-CoA synthesis decrease histone acetylation and will promote differentiation. But so the model that I'm using at the moment are the human trophoblast stem cells. So these cells can be either derived from blastocysts or from first trimester placentas, and then can differentiate in the two cell subtypes that I described before. So the syncytial trophoblast and the extravillous cytotrophoblast. But before talking about my experiment, I wanted to focus on how do we culture these cells. So in metabolic studies such as this one, the culture media is important because as it also been founded, like in these two studies, basically they come up with new formulation of the media that we use to culture our cells, which are respectively the human plasma-like medium and plasmex. So basically they formulate this media with a more physiological concentration based on the human metabolites, which are found in the human plasma. And we established a collaboration with one of these groups that gave us plasmex, so I could use it to test it on my cells. And so here I compared some metabolites which are found in plasmex and the common media DMEM-F12. So as we can see from this table, plasmex is a way more complex media than DMEM-F12. And if we consider that this is a logarithmic scale, some of the metabolites which are fundamental for the acetyl-CoA production, such as glucose and sodium pyruvate, are present in 3-fold higher or even 5-fold higher in the DMEM-F12. Other components, such as acetate and citrate, are not even present. So these non-physiological levels of nutrients in the DMEM can have an effect on cellular metabolism and studies based on cell metabolism. So I started to test these two media and compared the effect on HTSC cells. And as we can see here, when cells are cultured in plasmex, cell proliferation increases and increases also cell viability on the plasmex, which has been measured with the MTTSA, and we have a better basal respiration. Then I wanted to check what happens to histone acetylation and trophoblast stemness. So histone acetylation is increased and it is being measured through these acetylation levels of this protein, and trophoblast stemness doesn't change. So now that we know that plasmex improve proliferation and acetylation, I wanted to see what happens on differentiated cells. So I tested again this media and I measured the level of two differentiation markers for STB and two differentiation marker for EVT. And there are different time points where I measured these mRNA levels, and under plasmex, cell differentiations improve. So to summarize, CDB cells have a greater metabolic activity and proliferate more when in plasmex, and CDB stemness shows no alteration and histone acetylation increases. So based on this finding, I decided to use plasmex to perform all of my experiment that I'm going to describe you. So getting back to my project, the aims are to characterize the changes in metabolism and histone acetylation during trophoblast differentiation, examine what are the enzymes that are involved in the acetyl-CoA metabolism and histone acetylation and trophoblast differentiation, and then investigate the mechanisms which link histone acetylation with trophoblast differentiation. So the metabolites in the placenta have been measured as a whole, but there haven't been studies where they measure the metabolites in the cell subtype as we did here. So we measure different glycolytic intermediates, which are metabolites that can be found in the glycolysis, a pathway that metabolites glucose into pyruvate. And we can see that in some of them, these are higher in the cytotrophoblast cells compared to their more differentiated state as STB and EBT. We also measure the extracellular acidification rate, which is a measure of the glycolytic activity. And again, it was higher in the CDB type compared to STB and EBT. And as the product of glycolysis in the end is the acetyl-CoA, we measure also the levels of acetyl-CoA in total, and again are higher in CDB compared to STB and EBT. So this supports my hypothesis whether a greater glycolytic activity is usually associated with higher acetyl-CoA levels, which then are able to diffuse in the nuclei and perform histone acetylation and supports trophoblast differentiation. Here we wanted to have a closer look to what are the enzymes that are involved in the acetyl-CoA production directly. So we target ACY and ACS1 both in the nuclei and in the cytosol, and we wanted to check if there was a difference in the levels of these proteins, again, in our 3 cellular subtypes. And so as we can see here, ACY and ACS1 don't change in the cytoplasmic level of CDB, STB, and EBT, while if we look at the nuclear levels of these enzymes, they are higher in the CDB cells compared to EBT and STB. So this might suggest that the metabolic regulation of the acetyl-CoA is directly correlated to these enzymes, which would be the focus of gene targeting experiments such as this one. So here we focus on this enzyme, we transfect our cells with sRNA for silencing, we knock down this enzyme, and I measured the knockdown efficiency, measuring the levels of this protein. So we know that it works. So my prediction was then that, of course, when these enzymes are not available anymore, the acetyl-CoA levels will decrease, and these won't be available anymore to support histone acetylation and therefore trophoblast stemness. So what we found was that histone acetylation levels after knockdown of this enzyme decreases, and trophoblast stemness decreases as well. So these might support my hypothesis whether an enzymatic regulation of the acetyl-CoA influences histone acetylation and trophoblast stemness. So in the same conditions, so we're in an experiment where, again, we knock down these enzymes. The prediction was, again, for the acetyl-CoA to go down. But in this case, we measure the differentiation marker for both STB and EBT in stem cells conditions. And here we saw that the STB marker increases when these enzymes are knocked down, while the EBT marker doesn't change. So interestingly, this might suggest that if we have reduced nuclear acetyl-CoA level, this may lead to a premature STB differentiation. So to summarize, the CTB cells are associated with high glycolytic profile and high acetyl-CoA levels. And the enzymes that are directly associated in the regulation of these metabolites are ACY and ACS1, which are again higher in the nuclei of the CTB compared to STB and EBT. Indeed, when we knock down these enzymes, histone acetylation decreases, trophoblast stemness decreases, and differentiation starts to get in towards the STB lineages. So just quickly, what I'm doing at the moment is performing single nuclei sequencing to examine what are the changes in the trophoblast trajectory and see what happens when we knock down the ACY. So we isolate the nuclei from the cells, which are then pushed into a microfluidic device and embedded in a tiny oil droplets with a barcoded beads, so we can track our cells. And this is the way how then we can differentiate the different populations that might be in my cell model. Then I want to identify the source of the acetyl-CoA for histone acetylation, examine what are going to be the effect of ACY silencing and differentiation on acetylation marks, and determine by using ATAC-seq and ChIP-seq the effect of this silencing on chromatin and histone acetylation landscape. And so with this, I wanted to thank Irving, which has been an amazing supervisor all the way through, Steve for his constructive feedback, Dafina for single cell nuclei lessons, and Francesca for feedback, and our collaborators that gave us the opportunity to work with Plasmex, and of course the founders of my scholarship, the CTR and the University of Cambridge. And I'm happy to take any question at the end. Thank you.